All right, so moving on to chapter 3 now. Remember to go and watch through the entire chapter video or read the whole chapter, preferably both, of course. Don't rely on this for all of the information you might need for the exam. This is definitely not going to get you through it. So let's review the major concepts that were covered in chapter 3. Chapter 3 talks about the tools that microbiologists use to study microorganisms. We start off introducing the concept of the 5 I's. What are the 5 I's? And yes, you do need to know them. And there is a reason for the order that they're in, so know the order of them as well. Inoculation, incubation, isolation, inspection, identification. These are basically the steps that you would be going through in order to ID an unknown organism, which we will be doing in the lab. So inoculation, that literally just means that we're going to take some sterile media, which you should know what media is. Media is the stuff that we're going to be using to grow up our bacteria, right? So typically, a lot of stuff that we use in our lab to begin with is going to be, just nutrient agar or nutrient broth that can grow most things, okay? It's just a general growth medium. So we'll inoculate, we'll have sterile media, and we will inoculate that. That means taking a little bit of a sample and touching it into the sterile media, and then allowing it to grow. We have to do that by putting it in an incubator. Most things like to be warm when they're growing. It's just what they're used to growing at, usually body temperature. So we put it in an incubator to keep it warm to help it grow. So that's incubation. So letting it double and letting it grow, that's the incubation part. So now we've taken a sample. Let's say we swabbed the bottom of our shoe and swiped it across some agar and popped it into the incubator. It's incubating now. That's our inoculation. Swab onto the shoe, onto the agar, that was inoculation, and then incubation into the incubator, warm to grow. Isolation, now we're going to take that out, 24 to 48 hours later, and then pick up some individual colonies for investigation, okay? That's isolation. So trying to either pick individual colonies from that sample or diluting out the sample more so we can get to that individual colony level to study individual bacteria. A colony is like a little mound of growth that develops from one single cell. So that one single cell leading to that one colony, that's a pure colony. That colony is one organism that is multiplied up. So it should be a genetically identical organisms in that colony. We can take that one colony that we know is just single by itself, should be pure, and inoculate a new media that is sterile, and that would be forming a pure culture. One single species of organism will be growing in that culture. That is a pure culture. Okay, it's not contaminated with any other bacteria. Right, and then inspection, that's going to be doing the different tests, whether that is stains that we'll be doing quite a lot of in lab, or growing on specific kinds of media that tell you whether or not this organism can use this, ingredient of the recipe, or that ingredient of a recipe, or can it grow at all on this super salty media. Some things can tolerate the salt and some things can't. And then if it changes color based on whether that organism can metabolize certain things to a certain product that changes the color of the media, all of that. And then using all of that information that we got from the stains and the media and all of that, and combining it together to identify. That's the 5 I's. So going into the media first, starting with selective media. Selective media is media where only certain kinds of bacteria can grow. There might be some sort of additive in it that is unfriendly to the growth of like gram-positive bacteria. There's a lot of media that's out there that selects for enterics. Enterics are usually the bacteria that grow in your gut. Gut bacteria tend to be gram-negative bacteria. So we have a lot of media out there that allow for gram-negative. On the contrary, we have mannitol salts agar. Mannitol salts agar is a very salty agar, what it suggests. And not a lot of organisms can grow on that salty of an agar. Things that can, however, are things that can grow on your skin, like Staphylococcus aureus, or Staphylococcus epidermidis. These organisms are pretty hardy and can survive in high salt environments. The other things can't. So that helps grow certain things and eliminate others. We have differential media. Basically, most things can grow on it, but based on whether or not it can use whatever additive you put into that media, it'll cause a color change. So what we see with mannitol salt agar, going back to that, because it is both selective with the salts and differential. Basically, there is mannose sugar that can be broken down by bacteria. And if they can utilize that sugar, they will create an acid byproduct. And there is a pH indicator called phenol red in that media that is a reddish orange color. If you make acid, that reddish orange indicator will turn bright yellow due to the pH change. So that tells you that this organism can utilize that sugar. Things can still grow on it and not utilize that sugar though. So that will tell you differences there. So selective, certain things can grow. Differential, you can tell by looking at changes usually in the media, whether or not something can utilize a specific nutrient. Okay. Usually color change. Fastidious is a term for organisms that require specific growth factors. And a growth factor is anything that an organism would need to be provided to it in its diet. That would, be required for it to live. So a good example, I feel like, for a because we are fastidious organisms, we can't make everything that we need to live, believe it or not. So if you've ever heard of scurvy, then you know what I'm talking about. Scurvy basically is a disease that develops due to vitamin C deficiency. Has sort of really bad disease-causing effects if you don't get enough vitamin C. So, that's why they would bring like limes and stuff on ships, because they weren't getting enough vitamin C and they could develop scurvy. So we can't make vitamin C. We have to have it provided to us in our diet. That's fastidious. That's a growth factor. Vitamin C is a growth factor. All right, so now we're going on to isolating our organisms. We talked a little bit about that, taking this, sample and then breaking it down to be sure that it is a pure culture. We can use things like streak plates, where we literally just take a plate. You guys know this from the lab, hopefully. You take a plate and then flame it, and then and then flame it, and then and then flame it. This is really bad. I'm sorry, but you get the idea. Each time that you're doing that, you're not picking up more sample, right? You're going back into the other one. So you're diluting it out with your streaks and just streaking out these bacterial cells to try to get them individualized so that you can grow individual colonies and then study those individual colonies. That's the purpose of a streak plate, isolation. All right, another tool that we use in microbiology to study microorganisms is the microscope. You will be very familiar with your good old microscope by the end of the course. Microscopy, we have basically you'll want to know the different kinds of microscopes, the light microscopes like we use in our lab, the electron microscopes that literally shoot electrons at things and then create images based on bounce back, essentially. We don't use those in our lab. Dark field is using light to create contrast from a dark background and a light organism. So it works kind of almost the essential opposite of what we have in our lab. Fluorescence microscopy, using fluorescent labels to identify specific parts of cells. Confocal microscopy essentially creates slices of things in image. Then you can piece those together and create 3D images from them. There's a bunch of other ones. So the parts of the microscope, I'm not going to go into that too much here because I don't have the picture of it here, but also because you need to learn this in the lab. You need to have learned the parts of your microscope in the lab. I'm just saying. So, but yeah, definitely know how magnification works and how to calculate up magnification. That's something you need to know in both the lecture and the lab. As far as that goes, whatever objective lens you're using, it's the ones you rotate between, right? Let's say you're at the 4x objective lens. That's magnifying something 4 times what it normally would be. And then you have your eyepiece that you're looking into. In most of the cases, the eyepiece is going to be a 10x, and it says it on the eyepiece, guys. A lot of people don't know that, and they're like, how would I know to do the math? Because it says it on it, but that's how. So if you have a 10x eyepiece and a 4x objective lens, then you multiply them to get the value of magnification overall. So now we have a 40x magnification. So we have magnified something 40 times what its usual size would be. That's what we're looking at through there. The real image is created by the objective lens. That's the first magnification. So that's the real image. And the virtual image is created through the ocular lens. There's mirrors and there's another magnification step involved. That's the virtual image, okay? Oil, we use oil to reduce light scatter, basically as light goes through different media, whether that is the glass and then the air. Usually that's what we're talking about here. Oil has a similar refraction as glass does, so we put oil on that to minimize the light scatter associated with changing from glass to air to the lens. Now it's glass to oil, which is pretty similar to the lens, and that minimizes the amount of scattering you have of the light rays. And why do we want that? Because it gives a much clearer image. Okay. Stains, we use stains to give contrast, essentially, so we can actually see the cells, because otherwise when you look at cells without a stain, which you'll see in your motility exercise, it's very difficult to see cells without stain. All right, the stain, one of the, for a little bit, hold on a second, talk. One of the stains that we start off with will be the Graham stain. And this is probably the most important stain you'll learn about in this class. The Graham stain is going to help us distinguish between gram positive and gram negative. Now it's actually going to have meaning for you whenever we look at these things. It's going to mean more a little bit when we get into the prokaryote descriptions, but we'll start here. The Graham stain, this guy named Graham developed it. We put crystal violet. It's a purple dye. Crystal violet stain. It's the primary stain. It's the first stain that we use to stain is the primary stain. That's not supposed to be a secret or anything. Not a trick. So it gets stuck in the thick walls of gram-positive bacteria. We'll talk about what that means in the next chapter. But gram-negative have very thin walls. They have another layer, another membrane outside of that, but they don't trap the crystal violet in the walls. That's where the crystal violet gets trapped up, is in the actual wall itself. It's very thick in gram-positives. We add iodine to help work as a mordant, sort of like a like brick and making mortar for bricks, sticking them together. That's what mordants do. Okay, so any of the crystal violet that did get stuck in that wall is now going to be fixed there by that mordant, the iodine. Then we decolorize. That is using ethanol. Ethanol is just regular drinking alcohol. It's nothing special. Using it to rinse out the excess crystal violet that didn't stick into the wall, whether it's positive or negative. And give it a good rinse through. If it got stuck in there because the thick cell wall and the mordant, it will not rinse out with the alcohol. If it is gram negative, it will rinse out with the alcohol because it didn't get stuck with that mordant in that thick cell wall, right? So it's just going to rinse out. So it rinses right out. So now at this point, we will counter stain with safranin. So a counter stain is a stain that you re-add in after a decolorizer. If your cells are purple after all of that, then you're gram positive. You stuck that crystal violet into your thick cell walls. Congratulations, you did your gram stain properly and you have purple cells. They are gram positive, purple for positive. However, if you look at your cells at the end of all of this and your cells are red, congratulations, you did your gram stain properly. You have gram negative bacteria and they look red. You know, red, I feel like is pretty negative color, I guess, associated with negative, whether it's charge or, just mood in general, whatever. So there you go. That's how the gram stain works. And that's what gram positive means. And gram negative means it literally just means what color it stains in the gram stain. So one of the most important defining features of how to separate out bacteria. It's usually step number one. All right, next we have another stain called the acid-fast stain. This is really only going to identify Mycobacterium and Nocardia species. SPP means species, by the way. So it will identify several species that fall into that genus. Anything Mycobacterium, whatever, Mycobacterium sphygmatis, Mycobacterium leprae, Mycobacterium tuberculosis, all of those guys are going to be acid-fast positive. And some of those words might sound familiar to you, like Mycobacterium tuberculosis. That is because that is literally the causative agent of tuberculosis. And it is an acid-fast bacteria. They have a special wax in their cell walls called mycolic acid. It's very waxy and thick, and it makes them very hardy. We use carbonyl fusion, which sticks in to that, that waxy surface, but it won't stay in regular cells. So then we'll rinse with an acid alcohol. And if that carbonyl fusion stays in when we're rinsing with the acid alcohol, we will counter stain with blue to look at the negatives. But if it stays in, your cells will be fuchsia. reddish pink, okay? But if you're not acid fast, you're acid fast negative, your cells will be blue from methylene blue counterstain. So that's how that one works. With endospores, endospores are living things. They are not metabolically active. They are metabolically inactive. And we'll talk about more in the next chapter. They are made by bacteria to help them survive harsh conditions and protect their genetic material until things get favorable again. So we have another spore, another stain that can help us identify endospores. We're not going to get into the details here, but we will be doing that one in lab. So do be aware of the fact that you can stain for endospores, and it will let you know if your bacteria is an endospore producing bacteria. The bacteria that produce endospores, Bacillus species and Clostridium species. We'll talk about that. Don't worry, I won't shut up about it for the rest of the time. So that is that for the tools that we use in microbiology, and I will see you guys next time for the prokaryotes.